co-transporters are common in animal cells. Because the sodium gradient is established by sodium potassium ATPase. This is good for Monday. Right? So what I'm saying now is stuff you don't even know for the test. Remember back in animal cells, we talked about the sodium, glucose, kind of reverting, this is sort of like review the exam. A co-transporter helps cells that line the intestine to absorb glucose. Okay? The sodium gradient is established by an ATP powered pump. Remember the sodium, potassium, ATPase? This is what builds the sodium gradient, high sodium outside the cell, low sodium inside the cell. Then through the symporter, sodium diffuses down this concentration gradient and brings glucose from low into high concentration. So it's called secondary active transport. Remember that? Secondary active transport. Okay. And I said this type of co-transport using sodium powered by the gradient established by this machine is present in animal cells, but not in plants or bacteria. Right? Plants and bacteria use different co-transport systems. They use as their ion H+ ions. Remember, the mitochondria used to be a bacterium. So this makes sense. The symporter, the co-transporter, is using a proton gradient, not sodium, to import drag pyruvate from low into high concentration. Remember that? Okay? That's a beautiful test question. I'll ask you to tell me back on Monday. A nice little essay. Only bitches have to grade it. Okay. Where was I? Oh yeah, Fremont. Okay, here we go. So we're going to import the pyruvate into the matrix. Ooh, Keanu Reeves, the matrix, where the enzymes are the citric acid cycle. Okay. Now we'll look. This is not showing you the co-transport, but we'll see that later. It's just showing it's on the way in. On the way in, it's going to be catalyzed by an incredible enzyme called pyruvate dehydrogenase. It's actually an enzyme complex. It's got a complex. It's unsure about itself. Pyruvate. This is an interesting enzyme. Pyruvate dehydrogenase complex. I had a cousin that had a complex. He thought he was a chicken. I said, no, you're really a human. Weird guy. All right. If an enzyme functions as a dehydrogenase, what kind of reaction is it driving? Can you tell me? Remember, if you're pulling off electrons, usually the protons, if you're really pulling off a hydrogen atom, what kind of reaction is that? We pull off an electron and the proton follows it and it goes somewhere else. What is it? No. Oxidation reduction reaction. Okay, so when you see it, the enzyme says dehydrogenase, you know the reaction is catalyzing as an oxidation reduction reaction. Remember I told you when something's reduced, you can tell if you see two molecules, one's converted to another, you can tell which one's oxidized or reduced. The reduced one has the hydrogens attached. The one's oxidized lost the hydrogens. So it's being dehydrogenated. Right? So this is an enzyme that drives an redox reaction. Does that make sense? Okay? Damn, this shit's complex, isn't it? Okay. So now this enzyme, it's a complex. There's actually three enzymes working together. It's one of the larger enzymatic molecular machines in the cell. It's almost the size of a ribosome. This thing is so freaking big. Okay? And that's what this is symbolizing here. I borrowed this from the Molecular Biology of the Cell, the great textbook of molecular biology. It's about the 9th edition or something. One's like $200. I mean, new edition. I'm like, shit, $200? Okay. So it's got three enzymes. Three enzymes working in this complex. It does three things. One thing, it decarboxylates. Ooh, that's a nasty word. Pyruvate to yield an acetyl group. So here's the pyruvate, CH3CO, and there's that carboxyl. It pulls that off as CO2 and it's lost to the world. What's left then is the acetyl group. Remember that? Should I draw that on the board or you're good? Huh? No, okay. So it does that, okay? That's the first part of it. One of the members of this enzyme complex. The other thing is that It also oxidizes pyruvate. Pulls away electrons and protons, and it shuttles them to the NAD, to make an NADH plus H plus. So it strips away high energy electrons. Okay? So this represents a product of this process. High energy electrons are now stored here. They can be later used to power ATP production in chemiosmosis. And the third thing, actually it's number 3, it doesn't matter the order. The third thing it does, this complex, is it attaches coenzyme A to the acetyl group, which has been decarboxylated. Okay? Coenzyme A. Okay? Coenzyme, they always refer to it like this. If you look at the structure, it's a big RNA nucleotide. It's another one, again, almost all these activated carriers in life are based on RNA molecules. I talked about how the scientists believe the early version of genetics was RNA based. A lot of the components of metabolism are based on RNA molecules. NADH, FADH2, coenzyme A, ATP. These are all modified RNA molecules at the foundation of cell chemistry. A relic of that first RNA world. So it's doing all 3 of these things. Okay? And coenzyme A is another activated carrier. Can you tell me 3 activated carriers real quick for the exam? Give me 3 activated carriers real quick. NADH? FADH2? Give me another one.
That one.
Coenzyme A? What about the 4th one, the most famous one? ATP. Okay? They're carrying energy, either a high energy phosphate group, a high energy electron. In this case, coenzyme A, what's the nature of the energy it's carrying? The acetyl group. Delivering the acetyl group to the enzymes in the matrix. Remember, I told you the acetyl group is energy rich. So enough. If I sketch the acetyl group over here. So remember, here's pyruvate. Okay. On the way in, that enzyme breaks this bond here. That's CO2. That's lost to CO2 to the world. Okay? Here's the acetyl group here. Okay? And it attaches through a sulfur linkage to this molecule. Let's see, I'll make sure I do the right molecule there. Yep. So, and it attaches here. Here's the coenzyme A. So this is a shuttle. This is the activated carrier to deliver this high energy product into the citric acid cycle. So this enzyme does 3 jobs at once. It's an incredible enzyme. We'll see in the Krebs cycle there's another enzyme that almost does the exact same thing. It's almost the same enzyme. Okay. All right. The Krebs cycle. Oh, brother, here we go. Whew. These cycles are brutal, man. Wait till exam 2. I think exam 1's hard, wait till exam 2. Then if you think exam 2's hard, wait till exam 3. Then if you think that's hard, wait till the final. Woo! I'm glad I'm not taking this class. I'd quit now. No, never. Okay. So here's acetyl-CoA. The 2 carbons are labeled in blue, so you can track them. Okay. It shuttles this high energy product into the Krebs cycle. Okay, citric acid cycle, same thing. And it links the acetyl group to the first substrate in the reaction, oxaloacetate. You don't have to know these names, but we have to use them like, hello Mary, hello Bob. Okay? So it links the 2 together, and in the process, the coenzyme A is released, so I can shuttle in another acetyl group. And when this bond is released, this hydrolysis of CoA, coenzyme A, releases a lot of energy that powers this reaction series. So breaking that high energy bond, the CoA to the acetyl group, is a release of energy here to power this process. Remember, stuff doesn't happen for free, man. Okay? No such thing as free. Someone says they got a great deal for you, they're screwing you. Okay? You follow me, you go to heaven. Bullshit. Okay. Okay, so the CoA is hydrolyzed, big energy release, and the acetyl group's attached. So the oxaloacetate 4 carbons receives the acetyl group. Now you got a 6 carbon citrate or citric acid. That's called the citric acid cycle. Okay? All right. Now, we're going to rearrange this via this enzyme to another molecule, isocitrate, so it's an isomer of that, still 6 carbons. And now we're going to have our first oxidation reduction reaction, where electrons are stripped away. Now here are the carbons from the acetyl group that were just added. They're in blue. Okay? We're going to use this enzyme here to we're going to rearrange this, and then isocitrate dehydrogenase. Ooh, there's a dehydrogenase. That means what kind of reaction is going to happen? A redox reaction. It's going to strip electrons away from this and deliver 2 electrons to NAD plus to make NADH. And this NADH keeps them in high energy. That's a yield. It's going to later contribute to the high ATP production at the end of the pathway. Okay? So we got a redox reaction here. Okay? And one of these, I believe it's this guy here, this CO is released as carbon dioxide. So there's our second carbon loss from our sugar. Okay? So we've lost 2 now carbons. Okay? All right? But notice it's not the carbon from the original import, it's in the substrate. These will be lost to CO2 in the next round. They'll actually go through this process twice. But those are facts that we can worry about later. So we have a redox reaction. We've harvested some electrons. We've lost a carbon here, so now we're down to a 5-carbon product, alpha-ketobutyrate. Okay? All right? Now we're going to go through the next reaction involves another dehydrogenase, another redox reaction. More electrons are stripped away, and they're hydrogens that follow. So we've got more yield here, and another carbon is lost. This carbonyl is lost, or carboxyl group. Okay? So now we've lost another carbon. Remember, we started with 3 carbons of